Thank you for joining us for this ICER meeting on JAK inhibitors and monoclonal antibodies for the treatment of atopic dermatitis. We're going to give people approximately 1 minute to log into the Zoom call, and we'll be officially opening the meeting then.
Good morning, good afternoon, or good evening, depending on where you are. This is the official call to order for this session of CEPAC, and I would like to start by welcoming all of you. And as the process is usually with our meetings, the first step is to declare our conflict of interests. So at the beginning, every CEPAC meeting, we want to make sure that all speakers disclose any financial relationship with industry. At this point, I will ask individually every member of the committee and ICER to present their conflict of interests. All of us, we have gone through the ICER conflict of interest policies, and we're going to update them as we go along. Also, during the course of the session, I will ask every person who wants to render an opinion to also declare those interests. I will start by declaring my own interests. My name is Defteris Milonakis. I'm a researcher and physician at Brown University. My research is supported by NIH, and I have received funding from BARDA for clinical trials. My institution has made contracts with companies to evaluate their clinic and perform clinical trials for them. This is a communication and agreements between the institution and the companies. I will mention those companies. Those are Regeneron, it's Cyclone, it's Chemic Labs, and Pfizer, Kaleido, Sidara. All of those clinical trials, most of them are for COVID-19 related work. It's not the last 12 months, but an older conflict that I would like to mention because it is pertinent to this discussion is a clinical trial that we did with Sanofi that ended more than a year ago. Thank you, and then I will go and start with each individual member in the order that they're listed here. And the first is Robert Asseltine, please.
Good morning, my name is Rob Asseltine. I'm professor and chair of the Division of Behavioral Sciences and Community Health at the University of Connecticut Health Center. I'm a medical sociologist, and I have no conflicts to declare.
Kelly Buckland. Austin Frank.
Hi, good morning. I'm a health economist and a researcher with affiliations at the Department of Veterans Affairs, Boston University, and Harvard University, and I have no conflicts to declare.
Thank you, Christopher Jones.
Good morning, Christopher Jones, Chief Ventures Investment Officer for the University of Vermont Health Network. I have no conflicts to declare relevant to this meeting.
Thank you. Rebecca Kirsch.
Good morning, Rebecca Kirch, the National Patient Advocate Foundation. No conflicts to disclose.
Thank you. Stephen Kogut.
Good morning. My name's Steve Kogut. I'm a pharmacist and a health outcomes researcher at the University of Rhode Island. and the Evaluation Director for Advanced CTR, which is Rhode Island's Clinical and Translational Research Program funded by the National Institute of General Medical Sciences. I have no conflicts of interest to declare.
Thank you. Donald Chris.
Good morning, everybody. I am Don Kreese. I'm the utility ratepayer advocate from New Hampshire, but I'm here today as a patient advocate because of my longstanding connection to the cystic fibrosis community, and I have no conflicts to disclose.
Thank you. Tara Laval.
Hi, everyone. I'm Tara Lavelle. I'm an assistant professor and health economist at Tufts Medical Center in Boston, and I have no conflicts to disclose.
Thank you. Kimberly Lenz.
Good morning, everyone. I'm the Director of Clinical and Operational Pharmacy for the Massachusetts Medicaid program, also an assistant professor in family medicine and community health at UMass Medical School, and I have no conflicts to declare.
Thank you. Greg Lowe.
Good morning, I'm Greg Lowe. I'm a pharmacist and pharmacoepidemiologist who works as the Director of Pharmacy Utilization for the Mass General Physicians Organization, and I don't have any conflicts in this subject matter. Thanks.
Thank you. Leslie Oaks.
Good morning, everyone. My name is Leslie Oaks. I'm a pharmacist and an associate professor at the School of Pharmacy at the University of New England.
Thank you. Jeannie Ryer.
Morning, everybody. I'm Jean Ryer. I'm director of the New Hampshire Citizens Health Initiative at the University of New Hampshire Institute for Health Policy and Practice, and I have no conflicts to declare.
Thank you. Jason Schwartz.
Good morning, everyone. My name is Jason Schwartz. I'm an associate professor in the Department of Health Policy and Management at the Yale School of Public Health. I have no conflicts to declare.
Thank you. Jason Wasfi.
Hi, good morning. My name is Jason Wasfi. I'm an associate professor at Harvard Medical School and a physician scientist cardiologist at the Massachusetts General Hospital. I am an academic physician. My research is funded by grants from the American Heart Association from the National Institutes of Health. And pertinent to the subject of this meeting, I have received consulting fees from Pfizer. So in that setting, I'm normally the chair of New England CEPAC. I have recused myself and very thankful to Dr. Malinakis for stepping in for me, and I will also not vote on the voting questions. Thank you.
Thank you. And Albert Whitaker, please.
Good morning, Reverend Albert Whitaker, and I am actually the Director of Community Impact for the American Heart Association, and I have no conflicts of interest.
Thank you. Now I will call on individual patient and clinical experts to introduce themselves and declare any potential conflicts of interest. Please note that Dr. Jonathan Silverberg will be joining us at 10 a.m. So we'll introduce any conflict of interest at that point in time. I will start with Dr. Elaine Siegfried.
I'm Dr. Elaine Siegfried. I'm a pediatric dermatologist for the last 30 years. I'm currently at St. Louis University's Cardinal Glenn Children's Hospital. I've worked with industry in a variety of ways, consulting honoraria, and especially as serving as the principal investigator for a variety of trials. And those companies include Incyte, Regeneron, Sanofi, Leo, Pfizer, and AbbVie. I also have received a fellowship funding, not me personally, but my institution, to support a 2-year fellowship training position for a fellow.
Thank you. Wendy Smith-Pekolka.
Hi, good morning, everyone. I'm Wendy Smith-Begulka, the Vice President of Scientific and Clinical Affairs with the National Eczema Association. As a salaried employee of NIA, NIA has received grants and sponsorship awards from a variety of industry partners, including the companies represented here today, which would include Pfizer, AbbVie, Sanofi, Regeneron, Incyte, and Leo Pharma. And I have also served on patient advisory boards for Pfizer and Incyte.
Thank you. And Samantha Bittner.
Hi, I'm Sam. I'm a patient ambassador for the National Eczema Association, but I have no conflicts of interest.
Thank you. Anyone else who would like to declare any conflict of interest at this point? Okay. Well, again, I ask that individuals declare their conflict of interest, so there is some transparency as we move along through the session. And now I will introduce Dr. Steve Pearson, President of ICER. Please.
Thank you, Eleftheris. And welcome, everybody. We start off our meetings by framing the question around why we are here today. And I wish we were able to have an in-person meeting. I hope that we are back to those soon. Our country continues to struggle with the best way to come together to share input and to help make decisions. The reason we're here today can start with this quote from a patient with atopic dermatitis. I think it's difficult for people to understand how living with eczema can vary greatly from person to person. Everyone's experience with eczema is unique and fluctuating. Eczema can simply be a mild annoyance to some. To others, it can be a debilitating condition that significantly reduces quality of life. The severity of one's eczema can wax and wane over time. Additionally, not only are there many different types of eczema, but there are also many different ways to manage and treat this condition. What works for one person's eczema might not work for someone else's. And so, as you can see here, one of the main reasons we're here today is because there is still significant unmet need for a condition that does vary a lot from patient to patient. And we all know how wide the variety can be in some ways of eczema. But I think we'll hear more today that severe atopic dermatitis can really have an important effect on people's lives and that of their families. So we're here today to talk about options for treatment for these patients and what we can do with new innovation. And the day that new treatments for a condition like this are approved by the FDA, what happens in some sense first is a sense of celebration. I mean, it means that we do have new treatment options. ICER had a meeting just a few years ago when Dupixent was the first biologic treatment to be made available. And here we are, just a couple years later, welcoming potentially several more new treatments to give patients and doctors new options. So there's a sense of celebration in the research, in the investment, and the risk, and the participation of patients and the research over the years. And that's an important reason that we're here today. We're also here, though, because patients from day one can have difficulty accessing these treatments. There are issues around coverage eligibility, questions that come up. There are questions and concerns about the costs, about what patients themselves have to pay out of pocket, and obviously what happens to their broader insurance premiums. And while we're going to be focusing on patients with severe atopic dermatitis, we also are here today because we hope to keep an eye on others in the healthcare system. And what happens to other people when new treatments are approved is sometimes harder to see, but still important. And I just want to share a couple of quick stories with folks who have been in the public media. And these are folks who have struggled in one way or another with issues related to rising healthcare costs overall. So Mr. Leonard Edlow, who's a pharmacist by training and still practicing in Virginia, also has led community efforts over the years to help bring witness and perspective to the struggles of people with their healthcare costs, out-of-pocket costs for drugs, but many other aspects of healthcare costs as well. Other stories include those like the family in Alaska, the Whitman family. This is a family with two young children, and in the story that was reported, they expressed, their story of having to drop health insurance because the premiums that they were paying as self-employed adults was getting too high for them to be able to afford. And the subsidies weren't enough for them to be able to afford the state exchange. So going without insurance is still, unfortunately, a problem for millions of Americans. And this family told stories of how they hadn't felt able to take their kids to the emergency room because they didn't have insurance. The kids had suffered concussion in one case and an injured leg in another. So the parents were really worried and upset, and they knew they were running important risks, but they felt really caught in a system in which they felt they couldn't afford the healthcare insurance that was made available to them. And lastly, on the far right, the McCoo family in Minnesota was in the news recently as one of what could be millions of examples of Americans who struggle with medical debt. There are 21 million Americans, according to a recent report, who are in medical debt and in collection. That means that they have been unable to make adequate payments on that debt. And this is obviously a very hard situation for individuals and for families. This family, their young daughter had cancer as a child, and throughout the course of that, the costs of everything, doctor's visits, radiation, hospital visits, other kinds of treatments, had led to significant medical debt. So we are here today, again, to really think about the importance of innovation and how we're going to welcome new treatments, new effective treatments into practice and use them appropriately. And we're doing that in a backdrop where we have to recognize that there are others in the healthcare system who can be, in some cases, severely harmed by the increases in healthcare costs that are driven in part by new treatments. So one way to see this just graphically is to think of our primary focus today on the patient and the family dealing with moderate to severe atopic dermatitis. We're going to talk about that, and we want to do the best for these patients and their families. But around them, around us all, we have to remember that there are other families who aren't in the meeting, if you will, but who are part of a broader community that we need to keep in our mind's eye and remember that they have important needs that we should also recognize. So it's in that hope of finding that right balance in part that we meet today to have a kind of discussion that we think is very healthy and important as we do welcome new innovation. So you've met the members of the New England CEPAC. I'm going to give you a little bit more background on ICER, the convening host for today's meeting. Our sources of revenue are kept updated on our website. And as shown here in this pie chart, the majority comes from nonprofit foundations. The chief of those is the Laura and John Arnold Foundation. We also take approximately 21% from a combination of contributions from life science companies and from health plans and provider groups. That is kept separate to help pay for an ICER policy summit and activities not related to the reports that are the subject of today's meeting. We have 10% from government contracts and grants and a small amount from other sources of individual philanthropy, et cetera. So the report today that's the kind of the backbone of the discussion started almost 8 months ago. And it started with what we call scoping. And that means we turn to patients and patient groups, clinical experts, the manufacturers in the field, and other stakeholders. And we talk to them extensively to get guidance on what are the key questions. From patients, we ask, what matters to you? What outcomes matter? What is captured in the clinical trials? What is not? What are the broader aspects of getting access and effective treatment that are important to you? And we really use that as our orienting framework for the entire effort that we undertake. Following that stage, we do an internal analysis of the existing evidence. That can be published evidence. It can be evidence that's been presented at meetings that we might be able to obtain from manufacturers under in-confidence procedures. It can be, in some cases, evidence that we are able to generate with patient groups themselves. But we do a very broad sweep to make sure that we're being as comprehensive as possible. The cost effectiveness part of our work is often done with outside academics. In this case, it was done with academics at the University of Washington. And the reports themselves, that each one goes through several cycles of public comment and revision with the key stakeholders and the larger public. In this case, we were also fortunate to have expert reviewers, Wendy Smith-Bagolka, who's here with us today, Dr. Silverberg, who will also be with us later, and Eric Simpson, another clinical expert. Inside the report, how it's structured to support the discussion and voting today is in some ways linked to our value assessment framework, which is a way of expressing what value is. And we often use another term, long-term value for money, which will be one of the voting questions later in the day. So value itself is obviously a complex idea, but in ways we think it can be kind of marshaled into conversation through looking at evidence and input on these different components. The first is, can treatments or other interventions help us live longer, give us health benefits by extending our life? They might do that, they may not. They may, however, return us to better function or improve our lives by having fewer side effects than other available treatment options. We look at cost as a part of value, and it's important to note that we're not just talking about drug costs, but all costs, both in the healthcare system, and the modeling can actually capture some effects outside in terms of effects on our ability to work, et cetera. So all of the costs are looked at as part of a comprehensive look at value. We will also talk about potential other benefits beyond health. These are things that are important to patients and families and to the health system and society, but may not be easily captured in the clinical trial evidence. And surrounding all of these in some ways, or framing them, can be special social or ethical priorities related to the severity of the condition, the magnitude of its effect over the lifetime course of the illness, and also our societal hopes to redress inequities in health and in other aspects of our society. So this is the broader frame that we hope to bring into the framework of our reports and certainly into the deliberation in our public meetings. And the agenda for today then will begin with a presentation of the clinical evidence, followed by the economic model information. We will have comments both from the manufacturers and from other public commenters, followed by a brief break. And then we will return to the further deliberation and votes by the New England CEPAC. We will have a lunch break, and then we'll come back after that for a policy roundtable. And just to give you an idea now of what that is, following the votes on the evidence, we will want to turn forward and think, how will this evidence now, how will it be interpreted by key stakeholders? How will patients and clinicians interpret it as they figure out what's best for them? How will insurers and others involved in health insurance, how will they look at it and translate it into insurance coverage, into out-of-pocket payment amounts, that kind of thing. And we'll also talk about the role that evidence plays in how companies set their prices and the efforts to gather further information on the benefits of treatments in order to reduce any uncertainty. So we'll do all that, close it up with reflections from the individual CEPAC members themselves, and target adjournment for 3:20 this afternoon. All right, with that, let me hand it over to Dr. Atlas, who will start with our presentation of the clinical evidence. Steve.
Good morning, everyone. I'm Steve Atlas. I'm a physician and health services researcher at Massachusetts General Hospital in Boston. Next slide, please. I'd like to acknowledge my collaborators on the evidence I'll present: Falusa Agbula, Grace Fox, Serena Heron-Smith, and Emily Nahn. These are my disclosures. I have no major conflicts. Next slide, please. Today, we're talking about atopic dermatitis, a common chronic condition affecting 11 to 15% of children, 7 to 10% of adults in the US. The annual cost is about $5.3 billion in the US, including over $1 billion in healthcare costs. Initially, atopic dermatitis can present with red papules and vesicles, pimples or bubbles with weeping, oozing, and crusting skin. Over time, these lesions can become dry, scaly, or excoriated with skin thickening and erosions, cracking and bleeding. The primary symptom is itching that varies in severity. Clinically, we measure severity based upon the amount and location of the skin involved, its appearance, and the subjective impact of its symptoms. Next slide, please. The impact on patients for patients with moderate to severe disease, it can have a profound impact on quality of life. The itch can lead to a host of additional problems, including skin pain and infections, as well as disrupting sleep. The sleep disturbance and daytime fatigue can affect performance, including that in school and work. All of this can lead to psychological stress, including loss of self-esteem, anxiety, depression, and suicidal thoughts. And not only can this impact the patient, but also the families, caregivers, friends, and relationships. Next slide. The current management for all patients, the skincare include the use of moisturizers and emollients, avoiding triggers such as heat or cold, low humidity, or known allergens. Prescription topical steroid creams are often used for short-term intermittent use, and maintenance with other treatments such as topical calcineurin inhibitors or phosphodiesterase 4 inhibitors. For those not controlled adequately with topical therapies, phototherapy or systemic therapies affecting the body's immune response are used. Short-term use of systemic oral steroids, often overused, or cyclosporin are common, with long-term use of oral methotrexate, azathioprine, or mycophenolate. More recently, dupilumab, an IL-4 receptor antagonist that's administered by injection, is now commonly used in this population of those with moderate to severe disease. Next slide. So the scope of today's review includes two populations, the first being those with moderate to severe disease, where we're going to be comparing the clinical effectiveness of 4 new drugs, abrocitinib, baricitinib, upadacitinib, and tralokinumab, with comparators to topical therapies, including emollients with or without topical steroids or calcineurin inhibitors, as well as dupilumab. For the mild to moderate population, we're going to be comparing the clinical effectiveness of topical ruxolitinib cream to either a topical vehicle, placebo, or other topical therapies. Next slide, please. So this presents the different drugs. Abrocitinib, baricitinib, and upadacitinib are oral JAK inhibitors, and the doses that we'll be discussing today are on the right-hand column. Tralokinumab is an IL-13 monoclonal antibody that is given by subcutaneous injection, with the prescribing information noted. In the mild to moderate population, we're presenting ruxolitinib cream, which is a topical JAK inhibitor, and the 2 different doses. Next slide. In terms of the key clinical outcomes, for the moderate and severe population, most of the primary outcomes are presented at 16 weeks. The exception is 12 weeks for trials including abrocitinib, and in the mild to moderate population, 8 weeks for ruxolitinib cream. In terms of the primary outcomes from these clinical trials, the Eczema Area Severity Index, or EASI, is the EASI response is the percentage improvement in this score from baseline. For example, we'll be presenting the EASI 75, which is the percent improvement from baseline that is greater than or equal to 75%. There's also the Investigator Global Assessment, or IGA. The IGA response is a score of 0 or 1, reflecting clear or almost clear skin, and can also include an improvement from baseline of 2 or more points. Other secondary outcomes from these trials include the Peak Pruritus Numeric Rating Scale, or itch scale, where we'll be presenting results where a greater than or equal to 4-point improvement from baseline. We'll also talk about other patient-reported outcomes such as sleep, anxiety, and quality of life, as well as harms from these trials, including side effects, discontinuation, and infections such as herpetic infections. Next slide. We had discussions with patients and caregivers, and they highlighted that most with atopic dermatitis have a milder course, but this may lead to an underappreciation of its profound effects on all aspects of a patient's life. Many deficiencies with current therapies support the need for new ones that work quickly, provide sustained relief, and are safe for long-term use. Concerns were also expressed about the high cost of care, including the need for multiple over-the-counter treatments, provider visits, prescription creams, and systemic medications. Next slide. Now let's turn to the clinical evidence. Next slide. So let's first talk about the moderate to severe population. We conducted a systematic review that's highlighted in the report. We identified 21 trials to include in a network meta-analysis. This included 15 monotherapy or placebo-only trials, as well as 6 combination trials that included placebo where the patient could also receive topical prescription creams. For the different drugs, we list the number of trials, the patients, and whether the trials also included younger patients, adolescents 12 to 17. So you can note for abrocitinib and upadacitinib, these trials included both adult and pediatric populations. And we also include trials from the dupilumab, one of the comparators that we'll be discussing. Next slide, please. This figure is a diagram of our network meta-analysis for the placebo only or monotherapy trials. Just to highlight this to it, none of the trials provided direct comparisons to each other. All of them work through placebo, PBO at the bottom. And all of the analyses are comparing them to placebo, and thus our analyses are indirect comparisons across them. The exception is in the lower right, there's dupilumab, which there is a direct comparison with the higher dose of upadacitinib in one trial. Next slide, please. These are the results of that network meta-analysis in adult-only patients, presenting the percentage of patients achieving the EASI-75 response that I mentioned previously. So for example, in these trials, placebo occurred in 12%, achieving the EASI-75. And as you go down across the different drugs, you can see increasing EASI-75 responses going up to 67% for the higher dose of upadacitinib cream. The confidence intervals are presented on the right, and the other comparator, dupilumab, the EASI-75 response from these analyses was 49%. Next slide, please. Turning to the combination trials, these were the 6 trials that included both placebo and topical therapies. One can see in this figure that there are fewer trials included. All of them again compared the active treatments to placebo. The one exception here was abrocitinib, where both doses were compared to dupilumab, again at the bottom right. So there's a direct comparison between dupilumab and abrocitinib. All the others work through the indirect comparisons using placebo. Next slide, please. So discussing broadly the NMA results of these monotherapy and combination trials, this figure presents odds ratios, where an odds ratio greater than 1 indicates that the therapy that we're studying is superior to placebo. One can note that all of the interventions had higher odds of achieving EASI-75 IGA response in patient-reported itch compared to placebo. The interventions in the monotherapy trials had higher odds of achieving these outcomes than interventions in the combination therapy trials, as shown in the figure. Interventions in the monotherapy and combination trials had higher odds of achieving the investigator-reported response, the IGA, than the patient-reported itch outcome, as shown here as well. Next slide. There were other important outcomes to highlight. The NMA results also compared interventions with dupilumab. Here, outcomes favored higher doses of upadacitinib and abrocitinib, and as mentioned, these were direct comparisons. Outcomes for dupilumab were generally superior in indirect comparisons with baricitinib and tralokinumab. When we looked at patient outcomes, including sleep, quality of life, anxiety, and depression, these all showed similar benefits favoring the interventions versus placebo. In terms of adolescents, we saw similar outcomes to those of adults for the trials of abrocitinib and upadacitinib that included younger patients. And finally, there was limited long-term outcomes, but these suggested continuing benefit of the interventions and no new safety signals that we'll talk about momentarily. Next slide. So let's switch gears and now talk about the trials for the mild to moderate population. Here, we identified 3 trials looking at ruxolitinib cream. 2 of them were comparing 2 doses of the cream with placebo or vehicle, and these did include younger patients as depicted here. And there was one smaller phase 2 trial where there was also a comparison of the higher dose of the cream to a mid-potency topical steroid. This trial did not include younger individuals. Next slide. This presents the outcomes of this mild to moderate population, comparing the vehicle placebo to the 2 doses of ruxolitinib cream. And this shows the percent achieving the outcome in the different trials, the range of them, for EASI-75, IGA, and itch. And one can see that compared to placebo, both doses of ruxolitinib cream showed improvement in terms of these outcomes. And this was in a dose-dependent fashion. Other patient-reported outcomes showed similar favorable results compared to placebo. The outcomes for adolescents were also similar to those of adults. There was limited data comparing the higher dose of the ruxolitinib cream to the medium potency steroid cream, and this suggested similar or potentially better outcomes. And finally, there was limited long-term outcomes, also suggesting continuing benefit of the interventions. Next slide. In terms of the harms from the trials, first, for the trials that included patients with moderate to severe atopic dermatitis, severe adverse events and discontinuations were uncommon and comparable for the active therapies compared to placebo. Conjunctivitis and inflammation of the eye, herpetic infections, and other adverse events were also similar among the treatment arms. For patients in the long-term combination trials, harms leading to discontinuation were uncommon and similar or slightly higher for the patients who were receiving placebo. However, one important note is that the oral JAK inhibitors that have been approved for other indications include black box warnings for adverse events, including potentially serious infections, as well as cancer and blood clots. In terms of the trials for patients with mild to moderate disease, adverse events were also uncommon, and these were similar or less frequent for ruxolitinib cream compared to the placebo. Next slide. Turning to controversies and uncertainties. As mentioned, we had no direct comparison of the new agents to each other. So we used indirect quantitative methods, and these are more uncertain than if there were head-to-head studies. And although the side effects of the oral JAK inhibitors in these trials were uncommon, there are worrisome side effects that led to FDA warnings on this class of agents for other indications. And as a result, the FDA's delayed review for all of the JAK inhibitors for atopic dermatitis right now, including topical ruxolitinib cream. In terms of tralokinumab, this works through a mechanism more similar to dupilumab than the JAK inhibitors, but it lacks long-term safety profile of dupilumab. And it's worth noting that patients with atopic dermatitis often have other allergic conditions, such as rhinitis or sinus infections and asthma, but it's not known how these new agents affect these other conditions. Next slide. In terms of other potential benefits, all of these drugs are new therapies that reflect an improved understanding of the underlying disease mechanisms. These new therapies may also improve quality of life, including social interactions with family, friends, educational achievement, and potentially work performance. And then finally, the oral JAK inhibitors for moderate severe atopic dermatitis may reduce patient and caregiver and family burden over existing therapies like dupilumab that are given by injection. Next slide, please. In terms of public comments we received to the draft report, highlight includes outcomes from the clinical trials we presented do not capture the full range of potentially beneficial consequences of these new therapies on life activities, including school and work. And Sanofi Regeneron disagreed with a statement in our draft report that given a similar mechanism of action, the long-term safety of tralokinumab may be similar to that for dupilumab, and this was revised in the updated report. Next slide. So in summary, for patients with atopic dermatitis, for the moderate to severe population, abrocitinib, baricitinib, and tralokinumab and upadacitinib improved outcomes compared to placebo. Compared to dupilumab, abrocitinib and upadacitinib were similar or slightly better, and baricitinib or tralokinumab as presented were similar or slightly worse. There were few serious harms reported with low discontinuation rates in these trials, but there's ongoing concern about the potentially serious harms of oral JAK inhibitors as we understood from their use in other conditions. In terms of the mild to moderate population looking at ruxolitinib cream, it improved outcomes compared to vehicle, and there were few serious harms reported with low discontinuation rates. Next slide. So turning to our ICER evidence ratings, based upon the evidence review for patients with moderate to severe atopic dermatitis, failing topical therapies, ICER rates the net health benefits of abrocitinib, baricitinib, tralokinumab, and upadacitinib as promising but inconclusive compared to topical therapies alone. This reflects the positive trial results for these drugs, but also the FDA concerns about the oral JAK inhibitors around uncommon but potentially serious harms. For tralokinumab, while it works through a mechanism more similar to dupilumab than the JAK inhibitors, it lacks the same long-term safety profile of dupilumab and also appears less effective than the most effective JAK inhibitors, so that some uncertainties about the balance of the benefits and harms remain. Compared to each other, our rating, insufficient, reflects the lack of head-to-head trial results and limited long-term experience. For abrocitinib and upadacitinib compared to dupilumab, our insufficient rating reflects the potential that these new therapies may well have greater efficacy than dupilumab, but also the concern about potential harms with the oral JAK inhibitors versus considerable data suggesting long-term safety with dupilumab. For baricitinib and tralokinumab, the comparable or inferior rating compared to dupilumab reflects both the indirect comparisons from the analyses and available safety data. For patients with mild to moderate atopic dermatitis, ICER rates the net health benefits of ruxolitinib cream as comparable or better to vehicle alone. This reflects the positive trial results for this new therapy, but also the knowledge that there is systemic absorption, and while less than when administered orally, the FDA has delayed review of ruxolitinib cream because of the broader safety concerns for JAK inhibitors. And finally, there is insufficient information comparing ruxolitinib to topical therapies at the present time. Next slide. So with that, I look forward to any questions you all may have.
Thank you, Dr. Atlas. I will start the round of questions. We'll have to go a little over the allotted time, but my question has to do with giving us your perspective on the data on adolescents. Not all studies included them. How can we, what is the safe extent that we can interpret data from adults to adolescents? Because you introduce it on the part on the improvement of quality of life for the whole family, but it was not part of all studies. Thank you.
Sure. So first of all, for two of the drugs in the moderate to severe population, baricitinib and tralokinumab, there have been no studies that have reported outcomes in individuals under 18. For abrocitinib and upadacitinib, the pivotal phase 3 trials did include a small percentage of patients in the adolescent range. These did show outcomes in academic and confidence data we were provided, showing that the outcomes were broadly similar to those in adults. We'll likely hear from our clinical expert, Dr. Siegfried, later, about the issues of giving drugs like the oral JAK inhibitors in younger patients and the potential risk and how one may consider those in adolescents versus adults. There are ongoing trials for abrocitinib in adolescent-only populations that seem to show similar results. And we know for dupilumab, they have previously published trials in adolescents as well as children showing benefit of that drug compared to adolescents. So broadly, we would say that the outcomes appear comparable in the adolescent population to adults. The issue of safety may have specific concerns in the adolescent population, particularly when thinking about the oral JAK inhibitors. Is that helpful?
Thank you. I would like first to invite our clinical experts if they have any position about the data that were presented by Dr. Atlas at this point.
I have a comment. I think it's very useful to see those numbers across the board. I think it's still very difficult to compare. It's almost apples and oranges with the details of those trials. For example, if you take a mild to moderate population that's getting topical treatment, even the definition of mild to moderate is much more based on lesional severity as opposed to total body surface area severity, and looking at numbers like a EASI-75, it's just very hard to compare that kind of efficacy across the board. So I would just suggest that everybody do that with a little bit of caution.
I think that's a great point. There are, as Dr. Siegfried says, the definitions of mild to moderate and moderate to severe are inexact. And there's limited data looking at sort of different groups, such as the benefits in severe patients versus moderate, as well as the different treatment options that are available. Each of the trials had unique aspects, and while we present the results in a comparative way, there are differences in severity, age, baseline scores that are not significantly different, but there are differences, including how they assessed outcomes. Broadly, they're very comparable, but Dr. Siegfried's correct that they are not identical.
Thank you, Dr. Atlas. I will move to questions from the CEPAC. I will start with Dr. Jason Swartz, please.
Thanks very much for that really helpful presentation. My question was about the safety conversation as well, since that did seem to shape some of the final evidence assessments, particularly the promising but inconclusive assessments. And it sounded as though the concerns about risks and long-term risks might have led to the promising but inconclusive grade, and perhaps not one of the other grades in the evidence matrix. Can you say a little bit, Dr. Atlas, about the JAK inhibitor safety story for those other indications that you noted? Are there things from those examples with respect to dosing or duration of use that could help us think about that safety context as applied to this condition? I'd be curious about that.
Yeah, so other conditions, primarily the oral JAK inhibitors are being used for rheumatoid arthritis. It's also an autoimmune condition, but we don't necessarily think that they may be the same as for atopic dermatitis. Clinicians also know that the severity of disease and the impact in rheumatoid arthritis may be different. So to say that the side effects seen in this other population applies here, I don't think we can say that. In current use, there's serious infections such as recurrence of tuberculosis, arterial and venous clots that can be quite severe. There's cancers that have been reported with it, as well as cardiovascular events. So these are rare, but potentially quite serious side effects. If one is facing a life-threatening condition or very severe disease, a patient may, in discussion with their doctor, say those risks are worth taking. But for a condition like atopic dermatitis that we don't think is a life-threatening condition, that decision, I think, really involves a shared decision-making process ultimately between patients and their physicians.
Thank you, Dr. Atlas. Dr. Lavelle, please.
Can I unmute myself? Hi, thanks for that presentation. I have a question about the comparators in this analysis. So for the moderate to severe patients, you compared the new agents or the new medicines to topical therapies and also to dupilumab. And so I'm curious about what is the appropriate comparator here in terms of, is dupilumab not appropriate for everybody? And what is the percentage of patients that are using dupilumab in terms of understanding, to set us up for the cost effectiveness analysis in terms of how to interpret the cost effectiveness ratios when compared to topical therapies as opposed to dupilumab.
Right. So specifically in the trials for moderate to severe, the comparator that we emphasize in our evidence is placebo. Now, obviously, that is not a reflection of clinical practice, and our data suggests that that may reflect the best, the most favorable outcomes for these drugs. When you look at the trials that also include the topical therapies, that would be more common how clinicians would use this, adding on top of topical therapies. There's also benefit that we see, though the magnitude is somewhat less. So in that range, we're going to be presenting the most favorable numbers in the cost effectiveness side that you'll hear about. In terms of dupilumab, it has become a very popular drug, a very favorable drug for those with moderate severe disease who are not being adequately controlled with topical therapies or other systemic therapies. It does have side effects. It doesn't work for everyone. It is given by injection. And I think the need for new therapies for those particularly who don't respond or may have side effects, conjunctivitis is one. New therapies, I think, are relevant, though this is a drug that has been very helpful for many patients.
And can I just ask one follow-up question? In comparison to the new medicines compared to each other, are they interchangeable, or is there continued, would there still be shared decision-making between the patients and the clinicians in terms of what is the right medicine for them?
I don't know if Dr. Siegfried may want to comment on this, but these drugs all have different potencies, as well as there's the oral JAK inhibitors, the injectable IL-13 tralokinumab. As I showed you in the data, the potency of these medicines do vary. The question of sort of being able to tailor them to the specific patient may be something that may come up. For insurers, they may think of these medicines as having fairly comparable outcomes, and we'll talk about that as well. The safety issues is also a relevant one that we don't know, is sort of is the less potent also have less side effects. We don't see any signals for that at present. Dr. Siegfried, do you have any comments about that?
Yeah, and I really do appreciate you're mentioning the role of the payers and lumping together safety and efficacy data based on very coarse measures. And I absolutely think that there are differences. It's important to have different tools in our toolbox for different patients. Even atopic dermatitis itself is not a single disease, it's a phenotype, and we don't have great biomarkers that distinguishes between them. And then even differences with a biologic that binds circulating IL-13, for example, is different than a biologic that binds the IL-4, IL-13 receptor. There's lots of pharmacometric differences in those. So you can't really compare them across the board. It is absolutely important to have more than one tool in our toolbox. And I think only time will help us tailor individual treatments for individual patients.
Thank you.
Thank you. Dr. Wasfi, if you allow me, I would take your question right after Dr. Brower's presentation, so as to try.
Of course, thank you.
Everyone on time. Dr. Brower will present the evidence and the economic modeling, please.
Hi, good morning. My name is Elizabeth Brower, and I'm going to be presenting the effectiveness and value portion of the report. Next slide, please. Before I begin, I'd like to acknowledge my team at the University of Washington, Josh Carlson, Elin Chen, and Ryan Hansen. Next slide, please. The objective of this portion of the report was to estimate cost effectiveness of abrocitinib, baricitinib, tralokinumab, and upadacitinib compared to topical emollients and dupilumab. Next slide, please. To give you a brief overview of our methods, we used a Markov model with a target population of adults with moderate to severe atopic dermatitis. It should be noted here that we assumed that adults over the age of 50 with a high risk of cardiovascular events were not included in this assumed target population. This is important as we assume that no therapies in this report have an impact on mortality, and JAK inhibitors are generally not indicated for that population due to potential complications. And we'll come back to this assumption throughout the presentation. We conducted the model assuming take place in the United States from a healthcare sector perspective. And we chose a time horizon of 5 years. We did that as compared to the standard lifetime time horizon in ICER reports for a couple reasons. The first being that we do not have data to model long-term outcomes of the included therapies. We also felt that a shorter time horizon would more accurately capture the patient experience with a single line of therapy. And we wanted to focus on a single line of therapy to keep the analysis transparent and straightforward for the included drugs. Finally, a lifetime time horizon is not as important if there is no assumed impact on mortality. We do, however, include a lifetime time horizon as a scenario analysis. The costs and outcomes in the model were discounted at 3% per year, and we chose a cycle length of 16 weeks to reflect the clinical trials. Our primary outcomes were cost per quality adjusted life year or QALY gained, cost per equal value life year gained, and we also included a cost consequence analysis of patient reported outcomes for itch, sleep, anxiety, and depression. Next slide, please. This slide includes both the model schematic and a few key assumptions of the model. I included them side by side so you could reference the schematic as I present it to you. I'm going to start by describing the schematic. So you see we have two main health states in our Markov model. The first is the no response or standard of care health state, and the second is the responder health state. In the no response health state, we assume this is where patients start at baseline and where they go when they go off treatment. And in the base case, the standard of care is assumed to be topical emollients without the use of topical corticosteroids. In the responder state, we have 3 levels of response as measured by the Eczema Area and Severity Index, or EASI score. Responders were defined as patients who achieved an EASI 50 or higher with 3 mutually exclusive categories of EASI 50 to 74, 75 to 89, and 90 and above. I'm now going to be moving to the bullet points. Transitions to the response state occur after 1 cycle, and patients do not change response levels after the initial response while on treatment. As patients discontinue therapy for whatever reason, they are assumed to return to the standard of care or no response health state. After transitioning off treatment, quality of life and costs are assumed to be equivalent to a patient who is eligible for treatment but never treated. We also assume that among responders, discontinuation rates do not vary by responder level. Patients on only topical treatment or the placebo arms of the trials who achieve an EASI 50 or greater after the first cycle transition to the non-response state at a rate equivalent to discontinuation rates for the placebo patients in the relevant clinical trials. And finally, we do have a death state in the model schematic, which patients can transition to from any other health state during any cycle. However, we assume that atopic dermatitis disease and treatments do not affect mortality in this model. Next slide, please. We're moving to key model inputs now, and a note on these inputs. A large portion of this data was submitted as academic in confidence. So you will see blacked out boxes in the next few slides to denote that the data is redacted. These data will be available no later than 18 months after the report is published. And we're disappointed not to be able to show you this data and some of the model inputs, as it does make the slides less interesting to look at. We utilize the results of the multinomial network meta-analysis of placebo-controlled monotherapy trials to inform the treatment-specific transitions to each responder health state in the first model cycle. Although transitions by EASI level are redacted, we do list the total percent of patients who responded after the first cycle, and you see that upadacitinib had the highest total percent of responders, and baricitinib had the lowest percent. Next slide, please. We utilized treatment-specific discontinuation rates for the first year after the initial cycle, and then for all subsequent years over the model time horizon where data was available. Per cycle discontinuation rates were derived from extension trial data or long-term follow-up data, contingent on patients achieving a minimum of EASI 50 at their initial 16-week evaluation. Treatment discontinuation for any reason resulted in transitioning back to the non-responder health state. And long-term discontinuation data for atopic dermatitis patients was not available for upadacitinib. And in the absence of that data, we assumed a rate equal to the highest rate within class. Next slide, please. We included the costs for each individual therapy in the model where available, and we used SSR health data to estimate discounts from the wholesale acquisition price. Importantly, price information was not yet available for abrocitinib and tralokinumab, and no known corroborated analyst pricing was available for those therapies. Per ICER protocol, we used placeholder prices for those therapies based on other therapies within class. So for abrocitinib, we averaged the net prices of baricitinib and upadacitinib as the placeholder price. And for tralokinumab, we used the net price of dupilumab as the placeholder price, and we assume it's administered every 2 weeks in the base case. Placeholder prices will be updated in future versions of the report as that information becomes available. But it's important to keep in mind through the rest of the presentation that there's no truth behind the cost numbers for abrocitinib and tralokinumab. They're purely placeholders, and any results based on these numbers should not be taken as fact. Next slide, please. In addition to therapy costs, we included non-therapy healthcare costs for items such as outpatient visits. And we used annual direct medical cost estimates from manufacturer-provided data derived from IBM Watson MarketScan claims database. Claims were analyzed for the years 2011 to 2018, and costs were updated to 2021 US dollars. These annual costs include all non-drug direct healthcare costs. We also included one-time subcutaneous training and monitoring costs for the injectable therapies. And we did not find evidence of any serious adverse events occurring in over 5% of subjects among any of the clinical trials, and therefore we did not include adverse events costs in the model. Next slide, please. As I mentioned earlier, we included a cost consequence analysis in the base case to capture patient-reported outcomes related to itch, sleep, and mental health. Therapies were included in this analysis only if there was PRO data available by health state and therefore able to be used in the model. And pooled baseline estimates include all trials with a baseline estimate for each PRO measure from which the change in the PRO was measured. Next slide, please. And then finally, we have health state utility estimates. We derived pooled health state utilities for each health state from manufacturer submitted data. And we estimated utility values for each health state by combining these estimates from treatments with available data disaggregated by health state and weighting them by the number of participants in each study. Next slide. We'll now move on to the results of our modeling exercise. In a cohort of patients with moderate to severe atopic dermatitis who received a single treatment beyond emollients for up to 5 years, we found that baricitinib had the lowest drug and total cost at about $27,000 and $105,000 respectively. And again, this is over a 5-year time horizon. This is compared to upadacitinib, who had $151,000 and $220,000 as the highest drug and total costs, respectively. Another reminder that the drug costs for abrocitinib and tralokinumab are placeholders. For outcomes, abrocitinib generated the highest QALYs at 3.59, closely followed by upadacitinib and dupilumab with 3.51 and 3.47 QALYs, respectively. And I'll note that although upadacitinib had the highest overall percent of responders, abrocitinib produced the most QALYs due to having the second highest percent of overall responders and a lower discontinuation rate relative to comparators. Equal value life years were equivalent to QALYs in this exercise because of the assumption that there was no effect on mortality. And similarly, life years were equivalent across all therapies because of the same assumption. Next slide, please. We then have our incremental results versus our two comparators, standard of care using topical emollients and versus the treatment already on the market, dupilumab. Focusing first on results versus standard of care, baricitinib had the lowest incremental cost per QALY gained at $71,000 approximately, and upadacitinib had the highest ICER at about $248,000 per QALY gained. Both of these ratios were largely driven by the incremental costs in the numerator of the ratio. Versus dupilumab, only abrocitinib and upadacitinib were more effective, but abrocitinib uses a placeholder price and must be interpreted as a placeholder. And upadacitinib did not meet any common thresholds for cost effectiveness in this comparison versus dupilumab. Next slide, please. These are the results for the base case patient-reported outcome cost consequence analysis, available only for tralokinumab and upadacitinib versus standard of care. These results can be interpreted as the average change in each patient-reported outcome score from a pooled baseline over a 5-year time horizon. And then we also included the total costs over that 5-year time horizon for reference. We included this in the analysis in response to several conversations with patients and providers that underscored the importance of these specific attributes to the patient community when looking for an effective therapy. And we present the results as an average change for each PRO. However, because we could not identify a specific threshold or clinically meaningful difference in any of these scores by which to highlight a change. But to interpret this through an example, in the one PRO that we have data for multiple drugs, peak pruritus numeric rating score, or PPNRS, you can see that both tralokinumab and upadacitinib provide much more itch relief than standard of care as measured by this score, with upadacitinib providing more relief on average. Next slide, please. We performed a one-way sensitivity analysis to understand the impact of individual model parameters on our outcomes. And across all models, health state utility values were identified as the most influential model input. And I include a tornado plot of abrocitinib versus standard of care as an example of this sensitivity analysis. Next slide, please. We also performed a probabilistic sensitivity analysis to understand how the distributions of our model parameters would impact the probability that included therapies would be cost-effective across common cost-effectiveness thresholds. This table only presents PSA results versus standard of care. Next slide, please. Moving on to our scenario analyses, we conducted a total of 5 scenario analyses to test how plausible changes from our base case model choices would affect our conclusions. The first was to change from a healthcare perspective to a modified societal perspective. And in this scenario, we included productivity loss due to moderate to severe atopic dermatitis as indirect costs by health state. And these were derived from a workplace productivity and activity impairment questionnaire in the upadacitinib clinical trials. In this scenario analysis, drug and patient outcomes remain the same as the base case. So in this scenario analysis, the total cost increased by over 10 percentage points more for standard of care versus the therapies. And in the incremental results, the modified societal perspective decreased the ICERs by 7 to 22% across therapies evaluated. And dupilumab becomes cost effective at a $100,000 per quality threshold. Next slide, please. We also ran a scenario analysis where we extended the model time horizon from 5 years to a lifetime horizon. However, we still assumed patient only used 1 line of therapy, and then after discontinuing that therapy, they returned to topical emollients. In this scenario, incremental cost effectiveness ratios decreased by about 4 to 13% across therapies evaluated, and upadacitinib became dominated versus dupilumab. We then ran a model that allowed patients to use concurrent topical corticosteroids. We did this in order to reflect a handful of studies or trials where this was included, and also to reflect a potentially common real-world practice. The clinical team ran a fixed effects network meta-analysis from the studies that included topical corticosteroids to derive new initial response health state transition probabilities. And then we also added the cost of topical corticosteroids to the model. And we found that total costs increased in the combination therapy scenario for all therapies, but decreased in the standard of care arm. QALYs increased 2 to 4% across all therapies. And incremental cost effectiveness results were all nominally larger in the combination therapy scenario when compared to standard of care. And they increased by about 9 to 14%, but they remained in the same order of cost effectiveness and largely did not change any thresholds. One note was that upadacitinib became dominated when compared to dupilumab. Next slide, please. The model should be interpreted with several limitations in mind. First, differential effects of treatments modeled on conditions such as itch and sleep may not completely be captured by generic quality of life instruments. However, we found that available data did not support the use of treatment-specific utilities, and we included the cost consequence analysis in order to bolster this aspect of the model. Second, potential incremental effects of some of these treatments on quality of life in subpopulations of people with atopic dermatitis, such as co-occurring asthma or chronic rhinosinusitis, were not explicitly captured in the current model. The value of some of these therapies might be slightly different to these subpopulations. And third, serious adverse events that occurred in less than 5% of the trial populations were excluded from the model. However, we note that there are some rare serious adverse events seen from the use of JAK inhibitors for other indications that may occur in the atopic dermatitis population. And those would impact both costs and patient health-related quality of life. Next slide, please. We received numerous comments on our draft report regarding our cost effectiveness model. And to name a few of the bigger ones, Leo Pharma and the Sanofi Regeneron criticized the implied equivalence of long-term safety between the JAK inhibitors and the monoclonal antibody drug classes in the model due to the exclusion of serious adverse events, which is a noted limitation of the model. Pfizer thought that the assumed exclusion of patients over 50 with increased cardiovascular risk was inappropriate speculation on the treatment population. As previously stated, we chose to exclude that population as our model does not include a scenario where any therapies increased risk of mortality. There were requests from both Pfizer and Leo Pharma that led to the inclusion of 2 additional scenario analyses that can be found in the report supplemental appendix. The first was abrocitinib patients were evaluated after the initial 12 weeks on therapy rather than the 16 weeks to reflect their clinical trials. And that led to very small decreases in the ICERs akin to about 1% or less. The second scenario analysis was a proportion of tralokinumab patients decreased dosing frequency after 16 weeks on the therapy, and that led to a decrease in drug costs and subsequently the ICERs. AbbVie criticized the 1% discount rate applied to upadacitinib based on the SSR health information. ICER reviewed this calculation and confirmed that we accurately reflected the available pricing and discounting data and did not use any quarters of data where the net pricing in SSR health was above that of the wholesale acquisition cost in estimating the net price and corresponding discount rates. And ICER would also like to note that we welcome any manufacturer provided net price data to inform our models. And finally, AbbVie pointed out that the source of cost data in the draft report did not capture differential economic savings by health state. And they provided an internal claims-based analysis of health costs for atopic dermatitis by patient severity level, which was reviewed by ICER and subsequently incorporated into the model. Next slide, please. So to summarize our findings, we found that abrocitinib produced the most QALYs of the therapies considered, followed very closely by upadacitinib and dupilumab over the 5-year time horizon. Baricitinib produced the fewest QALYs. Compared to the standard of care with emollients only, baricitinib was cost-effective at a $100,000 per QALY threshold, and dupilumab was cost-effective at a $150,000 per QALY threshold. For upadacitinib, a discount of approximately 36% off the wholesale acquisition cost would be needed to achieve a $150,000 per QALY threshold. And the estimated net price is about $174 per dose, which is a 1% discount off the WAC. And then we do not present any cost effectiveness conclusions on abrocitinib and tralokinumab. Those will be finalized when their prices are available. Next slide, please. Are there any questions?
Thank you. I would like to start by asking Dr. Wasfi to pose his question that was held up from before.
Thank you so much. First of all, I just wanted to congratulate both the evidence team and the economic team on a tremendously rigorous process and really thoughtful report. So thank you. I had a question, I guess it's for Dr. Atlas, an effect basis about the evidence rating. So we're confronted with a situation here where there are rigorous short-term data demonstrating efficacy. This is the comparison of the JAK inhibitors to topical therapies alone. But there's also a legitimate concern about long-term adverse effects that has not been resolved. So I just could you comment a bit about, and I can see why you arrived at a P/I evidence rating for that comparison, but this is a sort of understandable problem. You never in pre-approval trials can know, either because there's not enough statistical power to detect side effects because of the nature of the trial, or because the time length of the trial is insufficient to see the development, for example, of a malignancy. So can you just comment on that tension? I mean, I definitely understand the difficulty here, but that is a tension that you sort of know something's effective in the short term, but then there's a conceptual, but powerful conceptual model suggesting there could be harm in the long term, and you simply can't evaluate it. But we still need to comment on relative efficacy and make clinical decisions and think about a value-based price. Nonetheless, we have to make these decisions regardless of that lack of evidence.
Why don't I jump in on that one, Steve? So thanks, Jason. I agree. This is absolutely a tension, and it comes up on almost every ICER report. There's evidence that new black box warnings happen on a whole bunch of drugs during the first 5 years after they're on the market. And I think when it's for biologics, it's either 25% or higher than 25% of new biologics end up with a warning in the first 5 years that you didn't know existed at the time you approved it. So it's a real issue that you're getting these drugs, they're doing something good, and yet you don't know everything that you need to know about them. And as we've looked at that at ICER, if you're talking about a rapidly fatal illness or even a not so rapidly fatal illness, it's easier to go, well, we don't know everything bad that it might do, but it would take a lot of bad to overcome the good we're seeing. When you're talking about a minor illness, you're not going to accept worries about mortality that you haven't proven don't exist. This is somewhere between those. Severe atopic dermatitis is a really bad disease, and yet people mostly don't die from it. I mean, you'll hear occasional stories of suicide, but mostly people won't die of atopic dermatitis. The JAK inhibitors are apparently worrying the FDA enough that they put a hold not only on the oral, but on the topical that has much less absorption. So we know there's a swirl out there of worry. And I don't think we totally understand what that worry is. We know that there's a worry about cardiovascular stuff. We know that there is known problems with clotting that isn't very great, but it's definitely there, that there are concerns about cancer and infection. And so we're left thinking, particularly for the two more effective JAKs, these look like great drugs if they turn out to be safe. And that's what that promising but inconclusive rating is meant to show. We just don't think right now we could know more than that.
Thank you. Thank you very much. This is a very important topic that I'm sure they will discuss again. I have 4 questions and 5 minutes to go. So we'll go in rapid fire. I will ask Dr. Jones to go first, please.
Thank you very much, and thank you for that helpful presentation. I noted, the industry criticisms on differential discount rates and so on, but what I haven't seen is, adequate consideration in the clinical trials for people of color and Asian populations who are more likely to have AD. Just the reassurances that that those considerations are made. I'm a health economist. I don't know how to adequately capture that, but I'd like to just invite perhaps clinical experts to comment on that, or those involved in this study to see whether there are some special considerations that should be made to make sure that those populations receive the access required and receive the clinical trial representation.
Hey, Chris, this is Steve just jumping in. That's a great question that I know will be teed up for our policy roundtable discussion with the clinical experts and others at the table, including the manufacturers. So let's hold on to that.
Thanks, Steve.
Thank you. I will move to Steve Coggett, please.
Thanks. I'll try to be brief. So my question is about the deterministic sensitivity analysis for abrocitinib, which used a placeholder price. And despite the uncertainty in the price, it looks like the variability around that price was only about 10%. In fact, doesn't even seem to include the possibility that it would be priced the same as the more higher potency options. So just wondering if you could comment a little bit around the decision-making around variation in that price. And I think perhaps it makes the ICER look a little more favorable.
Thank you for that question. I will note that in the one-way sensitivity analysis, which is the deterministic one, all variables were varied by 10%. So that might be producing the change that you are noting. I can't say anything about the price of abrocitinib beyond that.
Just wondering, 25 to 50% would maybe be a little more reasonable given the uncertainty in the price, and even in just consideration with how, maybe other intervals used in modeling?
Sure, this is Josh Carlson. I'll just also note that it's relatively common around prices to around 10%, but in this case, I think it's a fair point. We do try and urge caution in interpreting the price, just because it is just largely unknown at this point. So, but the swing, it probably drifts up, but we don't really know which direction it's going. So we just sort of adhere to standard practice on that one. But your point is noted.
Yeah, and we do have a voting question on the cost effectiveness or the value for abrocitinib, although I think in your presentation you said you weren't going to comment on the value given the placeholder price. So just want.
I think there's a voting question, but I don't think that again, just don't overinterpret that. It is a placeholder price. So that's the key point here, is we do not know the actual price of it.
Thank you. We can discuss once we have, David will discuss once the question is posed to us. I would like to go to Dr. Fracht, Austin.
Yes, thank you. I have a question about the patient-reported outcomes. Not being familiar with the baseline that these are deviations from, I wonder if we have a moment to hear from clinicians about how significant these changes are, the clinical significance.
Dr. Siegfried, please.
I agree with your concern. I think they're incredibly difficult to interpret, especially to, just try to make it a numeric score. Probably if you're going to look at one of the patient reported outcomes, it's the most important clinically. I think it's really impact on sleep. The other ones are certainly important, but much more individualized. So I would pay attention to that one the most.
Could you clarify which specific question, like which outcome you concerned about, what the baseline values are, or more about how much of an improvement is meaningful? Or both? I'll take that as a nod. So first, I came late and I apologize. I'm Dr. Silverberg. I don't know if I'm supposed to state all my disclosures. I have worked with every single one of the companies as an investigator, even in terms of trial design, other issues, and all of that information is publicly available. In terms of the baseline values, I think there are differences between the studies that have evolved in the trial landscape. We have seen for pretty much all of the studies post-dupilumab trials, baseline levels a little bit less severe across the board, not just in the PROs, but in the ClinROs as well. And the analytical approaches, though, are relatively comparable. I think we would all agree, looking at the PROs from based on past interpretability band studies, these are all moderate to severe patients, but with varying degrees, because as we see with many other diseases, there's a discordance between PROs and ClinROs. The investigator may rate the patient as moderate, the patient may have severe itch, severe pain, and other things that would now elevate their own perception of disease to being more severe. But across the board, at least when you look at the aggregate mean values or median values or LS mean values, these are populations with moderate to severe disease by all accounts. In terms of meaningful improvements, that's where we do have established MCID values for a lot of these tools, and some of those analyses were done, although I'm not sure that the MCIDs were particularly used. Those analyses, post-hocs, were used for the modeling purposes, but we have seen post-hocs presented for all of the assets showing that these really do have above and beyond improvements of these PROs better than what the MCIDs would be. So we think that these are meaningful changes. I would say, I agree with Elaine about sleep being very important in terms of overall health. From a regulatory standpoint, it's NRS itch, peak pruritus NRS itch that holds the greatest weight with the agency. And there's some debate about the best way to analyze it. Most of the studies have shown a 2 or 3 point improvement would be the MCID. The agency requires a 4-point improvement because they anchor around ClinROs, around the IGA scale. all of these drugs have shown massive improvements, whether you look at a 2 or 3 or a 4-point improvement in RS itch. So I don't think we have questions about their improvement on the itch or PROs, or that these are appropriate baseline severities.
Thank you. I would like to thank the clinical experts. I would like to, if you allow me to move ahead and with the process of public comments.